people can start connecting. So we're now online, and attendees can join, but we will not start the actual poster session until 1:30. But we start the session, we start broadcasting the session a bit earlier so that people can join. Otherwise, the first 5 minutes is just people logging in.
Is it possible, Sarah, to try how is the presentation mode?
Yes, you can try and share your screen.
How is it? Share
Probably
screen.
you use the wrong link, like
for example,
so,
I select.
Yeah, exactly. In the bottom, you can share. So now Lordina is sharing her screen, and Victoria.
No, sorry. But then I
wait. At a time.
No. I will cancel it. Wait.
But you saw it, right? You saw my screen.
Yeah, do it again, Lordina. So then you have your time, and then I do it.
Okay.
Do it again just in case.
I think it's this.
I guess we can all try to be sure everything is fine.
Can you see it now?
Yes. Yeah. And then please make it full screen. The slideshow.
Slide presentation, yeah.
Exactly like that. Yes, that's how it should be.
Great, stop sharing. Yes. Thank you.
You want to go ahead?
One of you has an echo on your sound, and we'll have to mute, but I can't guess who it is.
Yeah, so please also, when the others are presenting, mute yourselves so that you don't, so that we don't have echoes.
I can go ahead and try sharing my screen.
Okay. Exactly, and then you just have to go to slide share mode. Perfect. That looks great.
Thank you.
Karel, did you get hold of Rouven?
Stop sharing. Hello everyone, Tolulope Ayeyemi and DSR P-TRAP project. Currently,
yeah, so I was just calling Rouven, and he has some trouble joining, it seems.
Okay.
He says he pressed the right link, like for panelists.
Yes.
Maybe. Yeah. What did you say?
If it doesn't work, you can just send him the ordinary participants list, participant link, Sarah.
Yeah.
And then you make him a speaker.
1 second.
Hey everyone.
Rouven, we hear you, we see you. Well, we don't see you. We anticipate we might see you.
Oh, great. You're here.
You're here. Welcome.
Perfect. Great. So the other ESRs have just made a trial to make sure that they can share their screens. If you also want to do that, you can do that by clicking the share screen button in the bottom of the page. Yeah, that looks perfect. Thank you.
Just how do I stop again?
How do you stop? Yeah, you have to go to the top. So that's a bit confusing because when you start, you're in the bottom, and then you have to go to the stop to stop.
Okay.
sharing.
Yes.
So, but if there is, I can always stop your presentation. So, okay, we'll soon start. So you will have your 5-minute presentations, and then we will have 5 minutes of questions after each one. And then if we have time in the end, we can do some general questions to you. We have 1 hour until we have a virtual coffee break, each one in front of our computer. So we have already some 50 attendees joining in for this session. That's nice. I think we'll have a couple of more joining as soon as we hit 1:30, and we'll go into starting the presentation. So we've had a really big interest for this workshop. So your PhD is really on a hot topic. I think that must that's great for you to be on such a timely topic. And I hope you have enjoyed the other sessions, and that you will enjoy the session in the afternoon as well.
And to all participants, if you have questions, please use the chat function, and please send the questions not just to all the panelists, but to all the panelists and participants. And then there can be general discussion. And we will identify questions from the questions on the chat and put them to the speakers.
Oh, we are so very much on time. That's not always the case when you're in physical meetings. Okay, we have some 70 people in the audience now. That's good. So I will just wait until it's 1:30 to give people that we don't start ahead of time. So for those newly joined, this is the poster session, the virtual poster session with students, the early stage researchers from the P-TRAP project that was introduced already yesterday by Tolulope Ayeyemi, who is the scientific coordinator of the project. And we've had several speakers in the other sessions as well, introducing different parts or different aspects of this project that is spanning from trapping phosphorus from agricultural runoff to try and reutilize it as a fertilizer. So it's 1:30, so let's just start. We will hear from 5 of the P-TRAP early stage researchers. So we'll start with Victoria Barcala, that is at Deltares in the Netherlands. She's a civil engineer and did her master thesis at Delft, where she specialized on phosphorus recovery from agro-industrial wastewater treatment. So Victoria will speak about capturing phosphorus in drained agricultural areas. So please, Victoria, you have 5 minutes.
Yes, thank you very much for the nice presentation, Sarah. And I'll try to share my screen. Yes, so, okay. So Sarah very nicely introduced myself just now. I'm Maria Victoria Barcala, and I'm working at Deltares, and I'm also a PhD candidate at Utrecht University. And I'm working on improving the management of surface waters draining agricultural areas to optimize the retention and recovery of iron-associated phosphorus. So within the P-TRAP project, this is inside work package 1. that is to optimize the retention and recovery of iron-associated phosphorus. So from its application in the fields until it's discharged through the drains or groundwaters into surface waters. So in some areas with rich iron groundwaters, iron is usually reduced, so as iron 2, and it oxidizes as it seeps into surface waters. And in this process, that is what we can see in the picture, phosphorus can be adsorbed into iron-rich colloids or also co-precipitate with iron hydroxide. It's important to know how the hydrological and geochemical conditions are in agricultural areas to design good phosphorus retention measures. And that's what I will be working on. So on my first year, I have been working on previously collected the data by some colleagues, Deltares, and the idea is to study how is the phosphorus transport at the farm scale. And we selected a particular farm in the Netherlands where all the water drains through subsurface to one main ditch where we place a high frequency monitoring station. This data was also complemented by high resolution soil extractions at different depths, as you can see on the pictures. And also on the ditch, we can see the formation of these iron-rich particles. Also, this farm has iron-rich sandy soils, and manure has been applied here for many decades. As a result, we could do a phosphorus balance of the farm and also a water balance. And we concluded that although phosphorus is in balance with the phosphorus application in manure is in balance with phosphorus uptake by the crops, there's still a high amount of phosphorus leaching due to the high legacy phosphorus in the topsoil. However, due to the high iron content and amorphous iron in the soil, because I mentioned it was an iron-rich sandy soil, this iron absorbs the phosphorus and the phosphorus leaching is less than what it could be. Then in surface waters, the main transport is through the resuspension of particulate phosphorus. So this is good news because it means that water retention measures could be implemented together with particulate and phosphorus and particulate phosphorus retention. And this is good for farmers and may make them more likely to implement these kind of measures. Then for my second year, what are the plans? I will do my secondment as one of our partner organizations, that is Arcadis. And Arcadis is working on implemented iron-coated sand. That was a material that was presented yesterday that comes from a byproduct from drinking water companies. And they will use it to capture the phosphorus in full-scale big catchments. Here we can see one of the construction sites in Utrecht, that is before a lake in the Netherlands. Here we can see the outlet where they will use drains covered with iron-coated sand. So this will give us the opportunity to study full-scale measures and also to give scientific insight about how the iron-coated sand behaves under different conditions. And for this, I will do lab scale experiments, most probably with sand columns, and to study what happens with the iron-coated sand under reducing conditions, also higher pH, organic matter content, and flow velocities, that this might be one of the limits of the properties of iron-coated sand as a phosphorus adsorbent. And then finally, we will also address one of your questions that you mentioned yesterday, that was what to do with the iron-coated sand material when its sorption capacity is depleted. So study the quality of the material and what to do with it afterwards. These are the reference are used from Basis van der Grief's previous work. And thank you very much to all of you for your attention.
Perfect. Thank you very much, Victoria. Great timekeeping. So let's see if we have some questions from the audience. Chris, did you pick something up?
Yes, I guess you can see the questions, Victoria, in the chat anyway. So what crops are they growing on the farm in the Netherlands, and are they using cover crops to try and limit losses? And do you use ModFlow or other software to stimulate scenarios? So if we can maybe take those two questions first.
Yes. Yes, thank you very much for the interest and the questions. So the crops, 2/3 of the farms are grasslands, because also they have cows, yeah, like in a shelter. But this 1/3 that they use crops, it alternates between maize, potatoes. So it depends on the season, and they try to rotate the crops. But we also know that from the soil extractions that in the grassland area of the farm, the legacy phosphorus was less. So we think that because of the soil structure, this might be better for having less phosphorus losses, and yeah, so it's better for the soil. And then the second question, we haven't modeled it yet, but we were planning on using the Stella software to also see the long-term effect of this legacy phosphorus and how we would move either to surface water or to deeper groundwaters. And then another plan was to use SOVEC software that is made by Deltares to model the resuspension of these particles and be able to estimate how much are the phosphorus losses downstream. And then I see another question, that what the particulate phosphorus be retained. Yes, we'll have to see, but first it's a big basin, so it's like a sedimentation pond, and at the outlet we have the iron-coated sand. So also it's expected that most of the particles will first settle in the big pond and then go through the iron-coated sand filter just in the outlet. So I think that would be good for retaining the particles.
Okay.
And then how do you replace the iron-coated sand in such a structure when it's kind of reached the end of its useful life?
Yeah, that I think is one of the issues and would require further civil works. So also the idea is to see how we can improve also the maintenance and make the iron-coated sand last longer. So we have to change the material in larger periods of time. So not that frequently, also to make it cheaper and more efficient.
Thank you very much.
Great. Are there any other questions? Okay, we will move on to the next speaker. So thank you very much, Victoria. There might be more questions that we can take in the end of the session. So we move to next speaker, who is Lordina Eshun from University of Manchester in the UK. Lordina did her master at Wageningen University and has worked together with WETSUS, and now she will talk about microbial technologies. So the screen is yours, Lordina, for 5 minutes.
All right, thank you. Thank you for the introduction. So I'm Lordina Eshun. I'm ESL 5 and I'm at the University of Manchester. My topic is microbial technologies for converting phosphate-loaded Fe3 oxides. And within P-TRAP, I'm focused on using microorganisms to produce useful materials. And my specific aim is to develop a microbial technology that can transform phosphate-containing Fe3 oxides into useful materials that can be used as an agricultural fertilizer. So for my objectives, the first one is to identify some constraints on the conversion of phosphate-loaded Fe3 oxides to Vivianite. The second is to optimize the production of Biovivianite and to assess the effectiveness of the produced materials as an agricultural fertilizer. Yeah, so microorganisms have been known for several decades to be involved in biogeochemical changes on Earth. One of such is the microbial reduction of iron or manganese by iron-reducing bacteria. This, John talked about it in the previous session. So these microorganisms are able to couple the oxidation of electron donor to the reduction of ion, and this can result in the production of secondary materials such as magnetite. However, depending on the composition or the oxyanions present, we can have other minerals such as siderite, we can have green rust, we can also have vivianite forming. So presence of phosphate influences ion reduction by inhibiting the transformation to secondary materials such as magnetite. This is due to the blockage of the adsorption site of, for ion 2 caused by phosphate. Also, when we have phosphates in the medium, it can help to promote the formation of vivianite. This is actually based on the concentration of phosphate we have. So for instance, if we have low phosphate concentrations, we can have, and if you have low phosphate concentration and high concentration of carbonate, then we know that we are going to form siderites. In my project, I will try to optimize the absorption of phosphates on ion 3 minerals and monitor the subsequent bioconversion to vivianite. So this is just a graphical illustration of my work. And here I will try to, so here, okay, the first question is how effective or efficient are these model organisms able to transform the phosphate adsorbed on ion 3 minerals to vivianite, and how effective the produced material, which is vivianite, or other secondary minerals such as siderite, can be used as an agricultural fertilizer. So this is a preliminary experiment, like trial experiment, where we use soluble Fe3 oxides. And here we use soluble Fe3 oxides in the presence of phosphate, and we can see the biotransformation that has taken place. So in my experiment, I'll be producing bioreduced materials, and these will be characterized using electron microscopy techniques, SRD, and we will also use synchrotron-based radiation techniques, and this will be done through a secondment to one of our partners in EOWA in Switzerland. My experiment will also make use of waste Fe substrates, and therefore it is important to characterize the microbial diversity of the samples with DNA sequencing. For future work, I will try to, we will try to determine the effect of absorbed phosphates on the bioreduction of the Fe3 material, as well as the secondary minerals that will be produced. We try to optimize the experiments depending on the secondary minerals that will be produced, as well as the process itself. We will also make use of waste Fe3 substrates from industrial companies. And finally, a secondment will be arranged to Fertiberia and University of Seville. It's interesting that Fertiberia has already started these experiments. I really liked their presentation in the beginning. So it's going to be a great collaboration for me. And here we would want to test the effect of vivianite and other secondary materials as an agricultural fertilizer. So this is some take-home message. And the first one is that Geobacter or Schwannella are capable of producing secondary minerals from Fe bioreduction. The presence of phosphate also inhibits the biotransformation by blocking the sites for absorption of for Fe2. And finally, vivianite, which is a phosphate-rich Fe2 mineral, can form when we have the right conditions, such as we have reduced conditions at neutral pH, and also when we have right stoichiometry between ion 2 and phosphates. Thank you so much. Yeah.
Thank you very much, Lordina. I think we have a couple of questions for you from the chat. Chris, do you want to pick them up?
Yes, I had a question. You're working with a number of bacteria. To what extent might similar bacteria be found in soils or in sewage, for instance?
Sorry, again?
You're working with various bacteria, Geobacter, Schwannella. Are these the sort of things that you might naturally find in soil or in the sewage works?
Yeah, I know that we can find Schwannella and Geobacter in natural environment. Yes.
Okay. And then I'm trying to understand the questions that are in the chat. And maybe if you understand them directly better than I do.
So
one is kind of what sort of percentage can you succeed in orientating between Vivianite and Sediment?
oh, okay. So for the experiments I showed, actually I started the PhD. I think it's not that long. So the experiment was just a picture to show what I did before the lockdown. So actually I don't have any laboratory work done. Yeah. I just showed to like, it was kind of a trial experiment. So we didn't even analyze it yet.
Okay.
Okay, are there any other questions?
Yes, so I think that there is this question which is basically about if you use a material to remove phosphorus in a ditch or something, then you will end up with an iron to phosphorus ratio which is very high. Is that going to be compatible with producing Vivianite?
I don't know if I understood the question correctly.
I think it's if you're using an iron-based material as a phosphorus trap in a ditch or something, then you will have a very high iron to phosphorus ratio in the system, at least for the first few years until the system is saturated in phosphorus. So do you have a chance of producing Vivianite in those conditions? Because you indicated that having an excess of phosphorus was important for going towards Vivianite.
Yeah, so in my experiments we use so it's kind of like lab work, lab studies, and then we will try to use waste materials as well from companies. We are not going to use ditch. Yeah, but I think it's true that the ratio is important, and yeah.
So there's a kind of contradiction between having a lot of iron, which will be efficient in removing phosphorus, and having a low iron phosphorus ratio, which would push you towards Vivianite, maybe.
Yeah, I think that works, for instance, in the wastewater treatment plant. Okay, but my experiments, for instance, because I'm going to use waste materials and microorganisms, they sometimes get inhibited depending on the solid ion 3 that we have. So I'm thinking the microorganisms, so it actually depends on the activities of the microorganisms, if they're able to use or if they're able to bioreduce the solid Fe3 oxides. Because for instance, if you have higher concentration of Fe3, solid Fe3, microorganisms may not be able to bioreduce all of them, but with optimizing the system by giving them different electron donors and humics, it can help get some of the ion 3 reduced, and that can help to form Vivianite.
Okay, so effectively you adjust the conditions to develop the microorganisms you're interested in.
Yeah.
Yeah, okay. And then the last question, maybe at what range of temperatures are these bacteria active? Is it something that will work in all climates in Europe?
Yeah, so they are mesophilic, so yeah. But we also have some microorganisms that can work at high temperature conditions, like at thermophilic conditions. Yeah, there are microorganisms there.
Okay, that's great. I think that answers most of the questions. And if you want to reply to other people, via the chat, that's perfect.
Okay, thank you so much, Lordina. So next speaker is Tolulope Ayeyemi from University of Sevilla in Spain. Tolulope has a bachelor in agricultural science and did her master at the University of Bonn on soil fertility and P cycling. So we'll now hear on the effectiveness of Vivianite as a phosphorus and iron fertilizer. Please, Tolulope. Tolulope, are you on the mute? Okay, I think we can hear you now. It seems that your screen stops sharing.
Can you hear me now?
Yes, we can hear
see
you.
My screen?
Yes, so if you can just do it to full screen mode, it's perfect. Or slideshow mode.
Okay, I'm sorry.
That's great. Thank you very much.
Okay. Hello everyone, I'm Tolulope Ayeyemi and I'm ESR 8 of the P-TRAP project, currently hosted by the University of Sevilla in Spain for my doctorate program, which is focused on the effectiveness of Vivianite as a sustainable phosphorus and iron fertilizer for agricultural crops. So like you may already know that the P-TRAP project is a large and holistic one, but my task in this project is looking at the fertilizer aspect of it. And that is arranged in the work package 1, which is broken down also to closing the cycle of P application in agriculture, looking at the fertilizer aspect most especially. So a main objective of my work is to test Vivianite produced on different soils and crops and compare their yield and bioavailability with conventional fertilizers. So if we are going to introduce new fertilizers into fertilizer industry, we have to do enough tests to be sure that they can actually be used and accepted by farmers in different parts of Europe in soils and different crops. So I like to take a background on why this research is needed at this time. Probably we all know that there is a decline in phosphate rock quantity and quality, and this projected peak phosphorus, which is projected to be in the next 13 years, 2033, and also to talk about the fact that over time, the trend of world rock phosphate production and world population has been going at similar trends, and an increase in population of about 9 billion is also expected in 30 years from now. So therefore, we need alternative sources of phosphorus for crop production through recycling if we have to keep feeding human population. And another challenge I like to talk about is the challenge of eutrophication, which results from using of phosphate rock for producing phosphorus fertilizers, scarce resource, non-renewable, but then it still causes environmental challenges in some parts of the world. That takes me back to the concept of recycling and circular economy. A lot has been said about Vivianite in the morning section, but I'd just like to take a quick recap on what Vivianite is and what we know so far, and possibly what it can do in the future. So Vivianite can be found in reducing environment, and this first picture is actually found somewhere in the north of Cameroon, and it was used as a source of phosphorus on pod experiments involving the common beans, and the result was quite impressive. We also can synthesize Vivianite in the laboratory with a combination of ferrous sulfate and diammonium phosphate. This has also been tested especially to correct iron chlorosis in tree crops such as olive and peach. And the final one, which is most important for this presentation and workshop, is Vivianite can be precipitated from wastewater treatment plants, but we don't really have so much information on the behavior of Vivianite, especially as phosphorus fertilizers in different soils and different crops. So these are just general ideas and overviews, and from some of the presentation we also have this morning, but maybe these are not exactly enough information to introduce Vivianite as a fertilizer tool, especially for farmers use. So within the next 1 to 2 years, I will be working on some of these ideas, and I like to show you what my experiments and objectives are within this time. So right now I have a wide variety of Vivianite products from different companies, VETSUS, and also from Fertiberia, and the objective is to assess the effect of Vivianite on plant P uptake in 2 types of soils. I also will look into ion uptake. I want to test the impact of organic substances on the release of phosphorus and iron from Vivianite, and this is to identify microorganisms or microbes that could contribute to the mobilization of phosphorus and iron in Vivianite. The third is that I'll go beyond pod experiments or greenhouse experiments to testing Vivianite products under field conditions, and this is to assess and compare the effect of applying Vivianite as a fertilizer under pod and field experiments, especially where environmental conditions cannot exactly be controlled. The final thing is to test different Vivianite presentation on release of phosphorus and iron from Vivianite. So what I mean by the Vivianite presentation of form is in what form is it best in powder or in granules or in suspension, even as nanoparticles. Will phosphorus and iron from Vivianite be best released for plant uptake and use? So I like to end by saying that we have ideas or hypotheses. These are some of my hypotheses that Vivianite may have a potential to be used as phosphorus and iron fertilizer. It could act as a slow release fertilizer with reduced leaching potential. However, like I said, within the next 1 to 2 years, I would move from further experiment to field experiment and have some results. So I look forward to having opportunity on this platform to share my results with you. And I'd like to mention finally that I have a secondment as geoteams where I would be testing Vivianite, especially with farmers, group of farmers on their fields. So that would have less of research, as the case may be, but practical applications in the field. That will be in my 3rd year of PhD. Thank you for your attention, and you probably have some questions.
Thank you very much, Tolulope. I do think we have some questions from the audience. Chris, have you picked up some specific questions?
Yes, there's a question. It was somewhat what I was interested in. You said you would work on testing different Vivianites. What sort of differences do you expect between the Vivianites? Is it differences of impurities, I mean, other than the granulometry and the particle size, which you can obviously adjust by grinding and sieving, but the differences between different Vivianites, is it purity, is it crystallization, is it the oxidation level, or what are the differences between different Vivianites?
Okay, thank you very much. Like I said, we have, for example, I have 3 different products, Vivianite from VETSUS, and they have different levels of purity. So that's one of the things that is probably different from one product to the other. Then yes, we are also going to look at probably how the impurities or the Vivianites, different types, are going to also behave in the soil. So these are like a range of treatments that I have, and yes, that's the basic thing.
I had a second question, which is, are you still looking for other Vivianites? If some of the participants and registrants listening are producers of Vivianite, would you be interested in taking some of their Vivianite to test it, or do you already have too many different Vivianites?
I think we are still open to some, because right now I only have from 2 companies, VETSUS and Fertiberia. So if we have companies who are willing to give us samples as testing materials, I absolutely think that is fine.
Okay, well that's a very clear call to anyone who's out there. If you're producing Vivianite either deliberately or accidentally and would be interested in having it tested for its fertilizer value, then there may be an opportunity there. Someone is indeed asking, will any of the Vivianite products tested be from tile drainage? To which I guess the answer is, would you happen to have some samples of Vivianite from tile drainage that you could make available?
Well, within the P-TRAP projects, I think that focus is on wastewater treatment plants. So, but then maybe we would also look into that. But I guess I will have to confirm with my principal investigator and the whole P-TRAP project, as it case may be. I saw a question in the chat box which I like to quickly answer. It says, will you also do work on characterization? Yes, that's a first main part of my work, which I am currently working on, although it has been delayed by the pandemic situation. But yes, this is, yeah, I'm going to look into characterization. That's right.
Okay, I see a number of questions from people talking about Vivianite scaling in sewage works. So obviously that's a somewhat different issue, and we know there are similarities there. To some extent, the work on struvite recovery came from initially people working to prevent struvite precipitating in sewage works, and it obviously makes sense to recover something which precipitates easily and naturally. Do some of the people working on Vivianite scaling have some nice lumps of scaling Vivianite which could maybe be tested for their fertilizer value? Question to participants.
I also want to, I found a question in the comment box which says, am I planning to compare the fertilizers from struvite to Vivianite? Absolutely, yes. I have samples of Vivianite from aqua minerals which are included in my experiments. So, yeah.
Perfect.
Okay, great.
Thank you very much. Tolulope, please follow up if you see other questions. You can respond directly in the chat while we go on with the next speaker, who is Karel As from Bayreuth University. Karel has done master's studies in biotechnology and earth sciences at TU Delft and Utrecht. And we will hear now about lake restoration.
Okay, so hello everyone. So I will show you a bit about our idea on how to restore lakes using iron-coated sands. So this is, oh, let's go too fast. A second. Sorry about this. Okay, yes, so this is a project in close collaboration with Utrecht University as well. So Melanie Munch and Theodor Behrens are also close collaborators in this project. This would put me in the restoration part of the P-TRAP project. And in this case, we will try this using iron-coated sand. So the problem with lakes, with certain lakes which are internally loaded, is that even though if you decrease the external input and have a low inflow of phosphate, algae blooms might persist. And this is because of phosphate being recycled internally in the lake. So this means that reducing fertilizer usage is not enough. You require restoration of your lake. And one of the incentives to actually restore the lake is the European Water Framework Directive. Basically, all lakes should be restored to good ecological status. And in a survey for water managers, they found that 13% recognize a lack of investments in restoration measures as a potential threat to not achieve this status. So we would like to try it with iron-coated sands, which have been obviously much heard of already in this conference. And what we have is from aqua minerals with grain size 2 to 8 millimeters. And what we think is the main mechanism of action is that it prevents resuspension by sediment stabilization, so less flux from the sediment by resuspension, and sorption of PO4 by the iron on on the iron-coated sands. So first we checked so what is actually in this iron-coated sand? Well, in these bigger particles, actually only a small percentage, 12% is iron, more or less. And then there's some other metals like manganese, aluminum, and calcium and magnesium, but in much lower concentrations. So these are weight percentages, but mostly it's quartz. So what does this mean for our adsorption capacity? We made adsorption isotherm. So this is what you see on the right is an adsorption isotherm with on the x-axis the equilibrium concentration and on the y-axis the amount which is actually on our iron-coated sand. And we can see that in the beginning it has a high affinity, but it can't have so much phosphate loaded onto the iron-coated sand. And only after we cross a certain concentration, the number will increase very quickly, and here the binding is very efficient. So what kind of concentrations do we find in lake sediments? Well, above this 50 micromolars. So we predict that it will probably be quite efficient, this sorption by iron-coated sand. So the next step is, of course, to use it to see if this actually works for the lake sediment. And one of the main methods to do this in the laboratory is by sediment incubation. So these setups look like this. This picture is by Melanie Munch. And our test site is the following, the Große Brombaachsee, which is a mesotrophic lake, which doesn't sound too bad, but if it actually has to be restored to oligotrophic lake because this is the reference state. So we took sediments from here and incubated it and saw how much phosphate, how much ammonium would be emitted from the sediment. It's quite a deep lake, 30 meters, and it stratifies in summer. So in oxic condition, we found that there is the sediment acts as a net sink of phosphate and ammonium. And in anoxic conditions, we see an increase in both ammonia and phosphate. So at this point, we haven't really applied the iron-coated sand yet, but this is more our angle of where we are going with sediment incubation and how we will test the effectiveness of this material. So what do we see after in the sediment? We see that these reduced species and phosphate can be found much higher because of flux and prevention of oxidation of iron minerals and direct oxidation of your ammonium. So in the future, we will continue with this kind of work. We will test these iron byproducts in our sediment incubation setup. In a later stage, we might apply them in test sites. So possibly this lake we're talking about, and we will investigate the influence of different redox conditions on the stability of iron-coated sands. So therewith, I'd like to thank you for your attention. And these are all the people from the P-TRAP project, basically. So thank you.
Perfect. Thank you very much, Karel. I do think we have some questions for you as well.
Yeah, Chris Thornton, I have a first question. Can you just remind us where the iron-coated sand comes from? It's a byproduct of some industrial process, or?
Yes, I guess you could call it industrial. It's from the water purification plants. So they have these sand filters through which this water is going to be purified. And during this filtration, a lot of the contaminants, organic contaminants, and these kind of things, they are adsorbed to the sand, and iron ions are oxidizing on the surface of this iron-coated sand. And in this way, you get this byproduct, because this filter is, of course, doesn't have an endless lifespan. So at some point it has to be replaced.
So it's from drinking water treatment.
Yes.
And it contains iron, but effectively the organics are not being retained in the sand that you're getting.
We found with a loss on ignition approach, which is a bit rough of what you're actually releasing, it was about 10% organics of the mass. So it's significant, I would say.
Yeah, okay, so that is quite significant. I had a second question, then I'll come back to the questions in the chat. Sorry, I'm assuming a priority for the moderator, which I should not do. But when I started, there weren't any questions directly to you in the chat, so I had a good excuse. My other question was, you mentioned oxidation of ammonia at the end.
Yes.
Can you explain how that is related to effectively taking up the phosphorus? Because what you're doing is absorbing the phosphorus. You're absorbing available phosphorus.
We don't.
How does?
Yes. Yeah. Sorry. Yes, but we don't really expect ammonia to be adsorbed on the iron sands, because generally it would have also a positive surface, iron. So therefore, the affinity for phosphate would be much higher. So the question would be if it will not act as this kind of wall for ammonia. But this might be interesting to test to see if maybe other components can form interesting things together. Yes, so this is something which is a bit unknown, but it's good to take into account, because certain lakes might actually not be eutrophied because of phosphate release, but of ammonia release, of course. So I think it's good to also take into account the ammonia and not just phosphate.
Okay, right. So now I'll come back to some of the questions in the chat. One is, how would you apply the iron-coated sand in the lake? How would you distribute it?
Yes, that's a good question. This practical question, honestly, I haven't really addressed it, but of course, one of the disadvantages if you have just 10% iron compared to an iron salt would be that you require much more mass. But then again, it might be that iron-coated sand have a certain niche of application in which the ferric salts, because I also see here a question about the comparison with aluminum and ferric coagulant. I think that iron-coated sand could be a good option in lakes that are not so well buffered, because if you add a ferric salt, for instance, it will hydrolyze the water and might change the pH. And aluminum has can also have problems if pH becomes too low in certain lakes. Then this alumina, which is toxic, might be released. So I think there might be a niche for iron-coated sand in that sense. Badly buffered lakes suffering from resuspension, where it might be worth to indeed go to the lengths of applying so much mass to a lake. So I could imagine that there is this niche for iron-coated sands in this type of lakes.
Okay, right. And then there was a last question, which was about the phosphorus compared to the depth and the distance from the sediments in the water column, I guess. But you're essentially working on the sediment surface.
Yes, on the sediment water interface. Sorry, I didn't quite get the question.
It's the one from Ed Weinberg in the chat. Have you used the incubation column to measure the dissolved phosphorus and compared to depth?
Depth in the lake, you mean?
Yeah, the depth in the lake, the depth in the water column.
Yes. This also depends on the season. It changes a bit. In alignment with internal loading during the stratification period, in which there will be no oxygen in the bottom waters, the concentrations of dissolved phosphate are actually higher in the hypolimnion than in the metalimnion. So there is an effective flux indeed from the deeper parts of the lakes towards higher ones. So here you can see this buildup, but it depends on the season as well, how that matches, basically. Is that the answer to the question? Is that sufficient? Or?
We'll see if Ed posts further questions. Okay, thanks very much.
Thank you, Karel. We'll move on to the last speaker of the session, Rouven Metz. Rouven has a bachelor in geoecology and a master in environmental chemistry from Bayreuth, and is now at the University of Vienna in Austria. So please, Rouven, you can share your screen.
Yep. So yeah, it's again about Vivianite. I hope you don't get bored about it, but we are looking at the different level. So we want to look at the mechanisms and which influence the bioavailability of phosphorus and iron from Vivianite. So how can I get the next slide? Oh, no. So generally, we see a low bioavailability of phosphorus and iron in oxic soils, and organisms develop different strategies to increase this viability of iron and phosphorus. Basically, there are three mechanisms. So one is the proton promoted by exuding protons. Second one is ligand controlled, where organisms exude low molecular weight organic acids like citrate or specific iron binding siderophores. And these ligands form iron-3 complexes, and so they increase the solubility of iron-3 minerals. The third one is the reductive dissolution. So organisms exude reductants which transfer electrons to iron-3 oxides. So we get the iron reduced, and ferrous iron is more labile in the structure of the minerals, and it also has a higher solubility. And between the three mechanisms, we also see combined and synergistic effects. So if you look now on Vivianite, it's quite interesting because Vivianite is a hydrous ferrous iron phosphate. At the one hand, we have a very high phosphorus to iron ratio. And on the other hand, iron is present as ferrous iron, so in the reduced form. And also the structure of the mineral is quite interesting because we have these single iron octahedral groups, and these which are linked with the double iron octahedral groups, and they are linked by phosphate. So we get this kind of a sheet structure, and these sheets are just combined by hydrogen bonds. So this is a quite weak one. So Vivianite is just metastable under oxic conditions. So we get an oxidation of the ferrous iron to ferric iron, and then we probably get a disruption between these sheets. Yeah, so this is one thing which is quite interesting about it. So the question is, how will Vivianite behave in oxic soils? Because soils, we have like many factors which influencing probably the oxidation and also the dissolution process of Vivianite. For example, as the pH, the ionic strength, interfering ions as calcium or silica. We have phosphate sinks in soils, as for example, the roots or other minerals where phosphate could bind, and also organic ligands, as I explained at the beginning. And there's, of course, the unique substances which always not really sure what they are doing. And all of these factors will influence the formation of secondary mineral phases. And of course, with this, the bioavailability of phosphorus and iron from Vivianite. So what we are trying to do is to model these soil conditions in very controlled batch experiments, and so that we can understand the mechanisms and also the dissolution quantitatively. So what did we do so far? First, of course, we need the material. So we synthesized Vivianite in an unoxidized way. Next, we see a SEM image of our product. And then there's also already a problem, since we need to characterize our solid phase Vivianite. And since Vivianites get gradually oxidized, and so we find mostly amorphous phases, so we decided to go on with Raman spectroscopy, and we could, at least in the first study, find differences between, we could find the gradual oxidation of the Vivianite. We also started with first dissolution experiments in batch setups. So what we see is in the anoxic dissolution, we see a quite high solubility, which is around 10% of the initial material. But in the oxic dissolution is much lower. However, if we just scale it up, it's still quite high in comparison with iron oxides. And there we already saw like a very interesting effect of silica, which increased quite highly mobilization of phosphorus and iron under oxy conditions. We also did dissolution experiments under oxy conditions in the presence of organic ligands. And here we saw, first of all, quite interestingly, totally concurrent dissolution of Vivianite. So it stayed the same ratio between iron and phosphate, which is in the mineral. And we also saw like very interesting differences between the organic ligands. So for example, with citrate, which you can see as black lines, we saw very, very fast initial mobilization within a few minutes to hours. And also a formation of probably 2-2 complexes. Instead, the microbial siderophore, which binds specifically iron 3, mobilized iron much slower, but constantly. And we also saw a positive effect of humic substances. So a short outlook, what we are planning to do. So we will go on with very controlled batch experiments to simulate different soil solutions. And then the next step will be to go on with flow-through reactors to also get a steady state condition and also to control the phosphorus concentration in the effluent. And with these experiments, we try to parameterize our kinetic models to predict, like at different conditions, how much or how phosphorus and iron will be released or bioavailable. Thanks.
Thank you very much, Rouven. Interesting to hear how Vivianite is seen by microorganisms. So I believe we have some questions from the audience, I hope. Chris, did you pick up some questions?
I have one question I would like to ask. You mentioned siderophores. Can you tell us briefly what they are and what they do, and whether they could be maybe developed, used more, cultured?
You mean siderophores? Yeah, I think I explained it at the beginning. So these are ligands which specifically bind iron, and they form complexes so that the iron has a much higher solubility in solution. So so basically organisms could now take up the iron.
And so these are produced naturally by microorganisms or by plant roots?
They can be produced by microorganisms, but also plants excrete them.
And so citrate is a kind of synthetic siderophore, but it's not very well targeted.
Yeah, citrate is just a low molecular weight organic acid, which is also produced by some plants to also increase the iron availability.
And so the difference with the siderophore is that the siderophore is more targeted.
Yeah, exactly. It has a much higher affinity for iron.
Maybe Rouven, you can give again. You just mentioned in your presentation, but maybe you can mention again some other types of ligands similar to siderophores.
Sorry?
I think you listed earlier on in your presentation other ligands except for siderophores.
Yeah, ligands. You mean like this more lower molecular weight? For example, you could have not only citrate but oxalate are like the most typical ones.
Okay, do we have any other questions, Rouven?
The question there's a question on the use of citrate, whether you could use it to selectively extract Vivianite phosphorus from soils, I guess, in a kind of analysis rather than for fertilizer or agricultural use.
It might be possible, but of course, it's question like how it would behave like in the soil, because the mobilized iron won't be mobilized there forever. So it would probably again, like when it gets, that it will precipitate again. And also citrate is decomposed in the soil, so it won't stay there forever.
Okay. And then another question was, how does the solubility of Vivianite change with oxidation of the Vivianite?
Yeah, that's exactly what we try to figure out. There's currently an experiment running. But yeah, as we see, like the unoxidized Vivianite under anoxic conditions is much more, like tenfold higher soluble than in oxic conditions. So it seems that like under oxic conditions, we get, not sure, but maybe it's like a passivating layer. But yeah, we need to figure this out. It's also like a bit challenging, because if you look just with a XRD, you will find still pure Vivianite, even though you know that it's oxidized.
Yes, and that's obviously of relevance if you want to make it into some kind of agricultural product.
Exactly, because the solubility is changing over time.
Unless you're thinking of delivering it under nitrogen, then it's clearly going to be clearly going to be in oxic conditions. So that maybe means you might want to coat it in something, or because you could keep it away from oxygen by coating the Vivianite in a polymer on something.
Yeah, the question is if it's needed. So because as we see in the oxic experiment with the ligands, we found again a very high mobilization of these of Vivianite. So maybe like in the soil it's behaving quite differently. And with the soil you also have these sinks for phosphate. So probably also the formation of secondary minerals will be quite different. So yeah, it's a complex system. So, and there are many factors that could influence it. But yeah, we are trying to get a bit closer to it.
Okay, and coming back to the ligands, the siderophores, I mean, this obviously could open a market to develop biostimulant products where you could maybe, and humic acids as well, which seem to increase the availability of the iron phosphorus, to develop some kind of product that could be sold to farmers as a synthetic way of applying the ligands and the humic acids to the soil rather than the plants having to do it themselves.
I mean, yeah, there are commercial products of siderophores.
Okay, right.
As far as I know.
Okay, we've actually reached the end of the session. There are still a few questions. You're welcome to stay on in the chat. That will keep open for another 15 minutes or so, where you can continue speaking. And then the next session will start at 3 o'clock, where you will have to use the specific link for that session. So thank you very much to all the ESRs presenting, really great presentations, great timekeeping, all of you, very impressive. And as I said in the beginning, very timely topics of your PhD. Maybe not so timely in terms of other external events that struck right when you were starting, but I hope you'll overcome that.
Yep, so as Sarah says, the chat will be open for another 10 minutes. If you have questions for the speakers, do put them in the chat. Either the panelists will try and answer them now, or we will store them, because the chat is stored by us, and we will extract from it the questions and try and ensure that they're appropriately dealt with. And otherwise, see you back in just under half an hour.
Thank you.